Greetings again, dear students. It's Assistant Professor Dr. Ghaithali Jasim with the Nervous Physiology Part 2. We will start with a new subject, the transmembrane potential, so we can understand how the neurons develop the signal and how these neurons conduct the signal. So the transmembrane potential is the big title. The intracellular fluid and extracellular fluid differ markedly in ionic composition. The extracellular fluid contains high concentration of sodium ions and chloride ions, while the intracellular fluid contains high concentration of potassium ions and negatively charged proteins. We can see here an illustration of the cell membrane. We can see the interstitial fluid above, or extracellular fluid, which contains a higher concentration of sodium ions and chloride ions, while below we can see the cytosol, or inside the cell, intracellular fluid, which contains high concentrations of potassium and the large negatively charged protein. These are separated by the cell membrane. So because living cells have selectively permeable membranes, the ions are not distributed evenly. Ions cannot freely cross the lipid protein of the cell membrane, so they can enter or leave through membrane channels, and there are also active transport mechanisms that move specific ions into or out of the cell. This image here also, we can see that the biphospholipid layer of the cell membrane contains different types of channels which permit or selectively permits different types of ions. Here also we can see an illustration of the type of transport which is happening. We can see diffusion and facilitated diffusion, which needs a channel, and also we can see that there are active transport that needs a specific type of channel called pump and require ATP for their action. Another image, we can see here the cell membrane is separating the extracellular and intracellular compartments with high concentration of sodium outside the cell and high concentration of potassium inside the cell. So the passive and active forces do not ensure equal distribution of positive and negative charge across the cell membrane. The cell membrane refers to the structure that separates the inside of the cell from the outside environment. Embedded in the membrane are protein channels that permit certain ions, for example semipermeable, to cross through the membrane at a controlled rate. So the transmembrane potential results from combination of passive and active forces. According to the chemical gradient, because the intracellular fluid concentration of potassium is high, potassium tends to move out of the cell, and sodium, on the other hand, is an ion which has a higher concentration outside the cell, so it tends to move to the inside. According to the electrical gradient, because the membrane is much more permeable for potassium than it's to sodium, so potassium leave the cell more rapidly than sodium enter. Here we're talking about the number of the channels. Wherever there are larger number, for example, of potassium channel, this membrane is more permeable for potassium. As a result, the intracellular fluid loses positive charge and the extracellular fluid gains a positive charge. So it becomes more negative. We can see here in this illustration the movement of potassium ions through the semipermeable membrane from inside the cell, the cytosol, to the outside of the cell, the extracellular fluid, according to the concentration gradient. Here the symbol for the negatively charged proteins is the M negative. We can see here that the negatively charged proteins have a relatively larger volume, which cannot cross the protein channels. So the positive and negative charge are separated by a cell membrane, which constitutes to a resistance because it resists the free movement of the ions. So wherever a positive and negative ions are separated by a resistant, a cell membrane, a potential difference exists. We can see here in this image there is measuring of the transmembrane potential because of the unequal distribution of the ions. The transmembrane potential for a resting neuron is minus 70. The potential difference is measured in volts or millivolts. The resting potential or transmembrane potential is minus 0.07 volts or 70 millivolts for the neuron cell membrane in the resting position. Another factor is the electrochemical gradient. The electrical gradient can help or oppose the chemical gradient. For example, sodium in the extracellular fluid are attracted by the negative charge in the intracellular fluid, so both electrical and chemical forces push sodium into the cell, while the chemical gradient for potassium tend to drive them out of the cell, but the movement is opposed by, first, attraction between potassium, which is positively charged, and the negative charge inside the cell. The second, repulsion between the positive potassium and the positive charge outside the cell. The last factor contributing in the transmembrane potential is the sodium-potassium ATPase pump. At normal resting potential, there is a slow leakage of sodium into the cell and diffusion of potassium out of the cell. So the cell must use ATP to eject sodium and take back potassium. So sodium-potassium ATPase exchanges 3 intracellular sodium ions for 2 extracellular potassium ions. In this way, the pump contributes to the negativity of the transmembrane potential because each time the pump transports sodium and potassium, the cell is losing one positive charge. We can see here in this illustration the combination of both intracellular and extracellular ions, and we can see the movement of potassium and sodium, and here also we can recognize the action of sodium-potassium pump. Ion channels. Ions cross the cell membrane through membrane channels. There are several sodium and potassium ion channels called passive channels or leak channels because they are always open but can change permeability in response to a local condition. The cell membrane also contains active channels or gated channels. These are ion channels that open or close in response to a specific stimuli and can be in one of three states. Number 1, closed but capable of opening. Number 2, open, activated. Number 3, closed and incapable of opening. Classes of gated channels. Chemically regulated channels open or close when they bind to a specific chemical, for example, receptors that bind to ACH, acetylcholine, at the neuromuscular junction. Chemically regulated channels are the most on the dendrites and soma of the neurons in synapses. We can see here in this illustration the chemically regulated channel. We can see that if it's closed, the ion cannot pass. When the chemical binds to its specific receptor, the channel will open and the ion will pass. The second is the voltage regulated channels are in areas of the excitable tissue, a membrane capable of generation and conduction of the action potential. Voltage regulated channels open and close in response to change in the transmembrane potential. For example, the opening of voltage regulated sodium channels is the key step in the generation of an action potential. We can find in the middle here the illustration for the voltage regulated channel. We can see for the transmembrane potential of -70, the channel is closed but capable of opening. For example, in the transmembrane potential of -60, the channel are open, activated, and the transmembrane potential of +30, the channels are closed and incapable of opening. Last, the mechanically regulated channels. These open and close in response to physical distortion of the membrane surface. For example, sensory receptors that respond to touch, pressure, and vibration. We can see here on the right the mechanically regulated channels. When there is a pressure or distortion to the membrane, the channel open and the ions are passing through. At the end of this lecture, I want to show you this nice image about the channels and the ion distribution. We can easily recognize here that sodium has a higher concentration outside the cell, while potassium has a higher concentration inside the cell. We can see here different types of ion channels: sodium channels, potassium channels, and sodium-potassium ATPase pump. We can see here that this resembles an excitable tissue. Thank you very much. So this is the end of part 1, nervous system. If you have any question, please write to me on the academic profile.